This unit is covering what is called diagnostic media. Diagnostic media is used to identify organisms. It can also be used to test for water to see if there are coliforms in water. It can be used for a wide variety of things. And it is a very large section of microbiology. We're going to cover just briefly the basics of it. And the reason you do these diagnostic tests, you can see on the right over here, it's a different colored auger. Now, in the past sections of this course, especially on the section where it had the media microbiology, all the plates were kind of tan colored. The broth was tan colored. In fact, let me go back. The deep auger slant, what's called the deep auger tubes, the auger slants, those were all, that kind of color, this tan cream color media. So we're going back over here there's some right there. So they're that color. This is called general media. Anything will grow on it, just about. You can pick up any microbe. These types of media are not selective for anything. They're not differential. Things grow on it. It's there to support most organisms, and that's the color. Diagnostic media is going to have different colors, and that's what the entire process is based on, color changes. So the reason you would do this is you want to try to identify an organism. You're trying to narrow down the scope of what you're looking at and eliminate other organisms so you can figure out what your organism is. I gave you this. Here's the quiz for this in the video before I went through all that. But I'm going to start here. Here's the outline that is present under the study materials. So right there, and then also right there as well. So what these are on the left, these are just microbes that are part of a microbiology lab. You don't have to learn these, but you would use these type of microbes and run tests on them, and you can eliminate, okay, it looks like we have Proteus instead of E. coli, or we have Staph aureus instead of Staph epidermidis, that kind of thing. And then these are the types of media. So I wouldn't worry about this page, just an introduction, but we're going to start here first. So we're going to talk about some plates, and then I'll do another video on all the tubes and the slants and things like that. So this very first test, this is called, and this is what it would look like with microbes growing on it. This is an Eosin methylene blue auger plate. It's got a maroon color to it. It has these chemicals inside that can detect changes in pH, and therefore the organisms growing on the media will alter the color of the media they're growing on depending on whether or not they can do certain things. So for each one of these tests, for instance, this Eosin methylene blue auger, you have to know, what are you testing for? Okay, it is selective for gram-negative enteric bacteria, enteric meaning the gut microbes that are part of the mammalian digestive tract. Selective means it will only support growth of certain organisms. It's very selective. Differential means that the media will change colors depending on whether or not they can ferment certain substances. In this case, this media has lactose, a milk sugar. So it's differential for lactose fermentation and coliforms. Now, a coliform is a gram-negative rod that ferments that ferments lactose with acid and gas production in 24 hours. Coliforms also are lactose fermenters, but they look different on EMB than anything else. So what you would do is you would streak the plate. Now this is a video of a person. Here's the Bunsen burner. In fact, let's make it full screen, why don't we? So what is in their hand, I'm assuming, I got the sound off, but there is an inoculating loop. They have in this bottle, I'm sure, culture. So some bacteria growing. It looks like water, and then I actually see some diagnostic media in the background here. So this person has sterilized their loop. They are now, looks like they're going to open up this vial. He's going through some detailed explanation, but it's the whole aseptic technique. Yeah, little pinky finger is going to twist the cap off. Going to heat the barrel. You want to kill any contaminants that are there, keeping them from escaping. Now he's taking some out carefully. He's going to heat the barrel again. And the reason I'm showing you this, when you streak a plate, and that's what part of this diagnostic media is for. So look, he's taking his loop and he's still explaining blah, blah, blah. But now he's going to touch the loop to the auger, and he's going to, it looks like he's making, okay, just a small, okay. And he did. If he would have streaked it down the middle, that's a simple streak. He just made a little small area. You do that as well if you want to do little quadrants in your plate. And he put those microbes on there. And it looks like he's doing a streak for isolation. That's what he's doing. So he's now going to make another streak that goes into his initial streak like that. Yeah, but you see how he's going back and forth? That's how you streak a plate. So when you think of these diagnostic media plates, just think of this person taking their loop and they're putting microbes on the surface of the auger. So now that you saw that person, what he was doing, you would do the same thing with this. You would take a loop and you would make little streaks like this. Now this one was done on purpose to demonstrate all three of the possibilities. Over here it says negative lactose. So if EMV, now this is what it looks like when it's sterile. Okay, it's that maroon color. If basically there's no color change, then you have a negative. It's negative for lactose fermentation. It's also negative for being a coliform. This middle swipe is a little bit darker colonies. That's an organism that can ferment lactose, but it's not a coliform. And that's what you would look for, the dark. If you had dark colonies or these dark where the loop actually made a streak, that's positive for lactose fermentation. If you're wondering what's the big deal with that, well, only certain microbes can ferment lactose. So like I said, you do these tests to eliminate possibilities of what microbe you have, or maybe you're looking for a certain microbe in an environment or in water or something like that. See the green metallic sheen here? That's what coliforms do. Escherichia coli, E. coli, is a coliform. Coliforms come from the digestive tract of mammals. So these two up here are actually gram-negatives. You don't have to know that Proteus is a gram-negative. Staphylococcus is a gram-positive, and that's what grew on this side over here. This is Staph. This down the middle is Proteus, and then this over here is E. coli. So you should be able to identify, if on an exam, if you see a green metallic sheen on Eosin methylene blue, you need to be able to answer, what does that mean? Number one is a lactose fermenter, okay, because it did grow, and the green metallic sheen means that it's also a coliform. And coliforms are also lactose fermenters. The middle section is just showing a lactose fermenter, but not a coliform, and it has to do with how they metabolize their energy. Once again, you're doing this, it may not be the greatest analogy. These are like pregnancy tests. A pregnancy test plus a person's pregnant, negative, they're not pregnant. You're doing the same thing here. Let's say you had something, hey look, I found some E. coli in some water, and you streak it on an EMB plate and it doesn't grow anything, that means you don't have E. coli, right? If it grew that, then yes, you're right. You found some E. coli in somebody's water source. They have fecally contaminated water. So that's the whole basis of these tests. And the plates, you would just streak them normal way. And the plates, you can run all different tests for all different organisms. So let's go to the next one. This is Mac, or what is called Macconkey Agar. It looks like this when it's sterile. It's kind of a pinkish color like that. You don't really have to know the original color. You have to be able to tell what's a positive and what's a negative for the test. Macconkey Agar is also selected for gram-negative enteric bacteria, meaning only gram-negative bacteria in that category will grow on this. It is differential for lactose fermentation as well. Now, here is E. coli. It's definitely a stronger fuchsia colored. You can see it right there. So that's a coliform. This really is not differential for coliforms, but you can tell there's a difference. The one in the middle, see the color fuchsia, and I put up here what does a positive test look like? Fuchsia color means coliform, strong pink. So right there, that's a positive. Over here is a negative. So negative for lactose fermentation. These other two are positive. It's just another test you can run along with the EMB. Just a different kind of test, different colors. MSA auger plates. Mannitol salt auger. It has a high concentration of salt. Not all organisms can grow in salt. This one happens to be selective for Staphylococcus. And specifically, it actually will tell you the difference between staph species as well. It's differential for mannitol fermentation. Mannitol is a plant sugar. So if you have an organism that can break down mannitol and it can grow on this plate, it will turn yellow. So here is the original color of the media. Here's a plate, and that writing is very small, I'm sorry, but somebody streaked staph epidermidis, which is on skin, on this side, and they streaked on this side over here, the yellow, they streaked Staphylococcus aureus. And you can see the Staph aureus turned it yellow. The Staph epi, it pretty much stayed the original color. And that's what this is showing. Micrococcus luteus, obviously this is a negative over here on the right. In the middle, this would also be considered a negative. You see it didn't change the color that much. And what you do with these tests, it's all or none. And you see that yellow, that's a positive. Because originally it was a red color. This was some Staphylococcus aureus, which is also on skin. Person did looked like a streak for isolation on this, and it turned it yellow. Or in this case, look, it turned half the plate yellow. Just depends on where you put. If you put the staph in a little small area, that little small area would have turned yellow. So it's differential for mannitol fermenters. Once again, why is that important? Only certain organisms can break down mannitol. So you're doing, and when you don't just do one test. Diagnostic media could be 50 different tests, and then you go back after you let them incubate. And I didn't mention this, but all of these, of course, look, when you streak an EMB plate, it's like any other media. You would streak your microbe on there. You would then put it in an incubator, and it should say that somewhere. Read results within 24 hours. So all of these have to be read. It doesn't happen immediately. I mean, the microbes have to grow on the surface media for you to come back and read the color changes. And what you usually would do is you wouldn't do one at a time. You would inoculate 50 different types of media tests, and then you let them incubate 24, 48 hours, and then you would come back and just read all the results and compile your data from that. Staph aureus, yellow auger, Staph epidermidis, the original color. So this is a positive. These two would be considered negatives. For mannitol fermentation, basically. What's it negative for? Mannitol fermentation. Azide. You don't see this one very much. It's this color. It's kind of a color of general media. You can see on the left here would be a negative result. On the right would be a positive result. E. coli is a gram negative. Staph aureus is a gram positive. And this type of media, this plate, is selective for Staphylococcus and Streptococcus. So there's all the dark colonies, and on the other side, that's a negative. So on Azide, if you're trying to eliminate species, hey, look, I found some Staph aureus somewhere, and you streaked it on here and nothing grew, well, then you don't have Staph aureus or you don't have Staphylococcus. Blood auger. I think I minimized. I didn't. I had mentioned this. This is general media. There's a deep auger tube. We don't use that much. Here's a broth tube that's clear, sterile. There's an auger slant. There's a plate. When you're making sheep blood auger plates, you use the general media. It's either TSA, tryptic soy auger, or nutrient auger. Will grow anything. And at the end of the procedure, you add some sheep's blood. So 5 to 10% deferminated sheep's blood. So it's a red color because it's blood, and it has red blood cells in the agar. What you can do with this is you can test to see if you have a pathogen. If you have something that can break down red blood cells, then you have a very pathogenic organism. Some organisms can partially destroy or damage red cells, and then some organisms cannot damage red cells at all. So it's differential for hemolysis. In other words, hemo is blood, lysis is to dissolve. So it's differential for that. Gamma hemolysis here is a negative. You have an organism that you can see it grew on there, but it did not break down any red blood cells. Once again, the media, the majority of this plate is a general media. It'll grow just about anything. You're testing to see if this organism will break down red blood cells. And so, the middle one is alpha hemolysis. A lot of times it has a green color to it. Kind of has this green, that's not that green, moss green. That means that you have an organism that can damage or partially destroy red blood cells. And then beta hemolysis, you'll actually see on the sides when you hold it up to the light, you'll see a clearing where all the red cells were destroyed. And that right there is actually all the red cells are gone where they streak this. So that's beta hemolysis. So be able to tell the difference between the three of these. You have to be able to tell what is gamma, what's alpha, what's beta, and then go from there. So, okay, I'll start here next.